Hello viewers, welcome back to KIC'S tutorials. In our last class we talked about the cell. I introduced the cell to you, gave a brief history of the cell. We discussed the cell theory and the types of cells, and I compared prokaryotic cells and eukaryotic cells. And I told you that in this video we'll be talking about Mendelian inheritance, principle of dominance. So Mendelian inheritance has to do with inheritance according to Gregor Mendel. But before I go into that, I'd like to define genetics once again. Genetics is the branch of biology that is concerned with the study of genes, the genetic variation between individuals, and heredity in organisms. So Gregor Mendel is regarded as the father of genetics. That is because he laid the foundation for the study of genetics. He studied inheritance of traits, that is the patterns in the way genes or traits are handed down from parents to offspring. He worked mainly on the garden pea, with scientific name Pisum sativum. As the garden pea. Okay. As we know, peas are in the family of legumes. So he worked on the garden pea, and you might want to ask, of all the plants or of all the organisms that exist in the world, why did he choose garden pea? He chose garden pea for a number of reasons, which I will be stating. First, okay, in his time there were various varieties of garden pea that were available for him to use. There were up to like 34 varieties that he could study. So that was the first reason that he chose garden pea for his experiments. And then secondly, garden peas are easy to grow and hybridize artificially. Okay, what this means is that the garden pea naturally is self-fertilizing. That is, you have the male and female parts in the same flower, in the same plant, so they can fertilize each other in that flower. And you can also hybridize them artificially. What that means is that if you want to cross one plant with another, you can also do that. You can get the pollens from another plant using a brush. You brush them onto the stigma, that's the female part of the other plant. That is artificial hybridization. So the garden pea was ideal for both self-fertilization and artificial hybridization. Also, the garden pea has a short life cycle. So it was easy for him to observe a lot of generations in a short period of time. They didn't have to spend so many years studying the garden pea. So that helped him gather a lot of information from different generations. Okay, there are a number of other reasons, but I'll just be sticking to this because of time. Okay, that brings us to the characteristics he just discovered in the garden pea. In the course of his study, he discovered that there were 7 pairs of contrasting traits or characters in the garden pea, which are 1, flower color, 2, flower position. Okay, sorry, let me take this to the left so that we can have space. flower color, flower position, the stem length, seed shape, 5, cotyledon color, that the seed leaves, 6, pod shape, and 7, pod color. Okay, so these were the 7 contrasting characters he discovered in the garden pea, and he saw that in each plant is either the plant has let's say the flower color has, okay, let me explain it this way. For flower color, he discovered that he had purple or white. So a plant will either have purple flowers or white flowers, no intermediates, just these 2 extremes, purple, white. And then for flower position, they were either axial or terminal. And then for stem length, you either have a short, okay, a tall or short stem. For seed shape, he had either round or wrinkled. So one plant cannot have different seeds, some round, some wrinkled. You stick to either round or wrinkled seeds. Then cotyledon color had either yellow or green. And then pod shape, these are the shape of the ripe pods. It either had inflated or constricted. And then pod color, either had green, those are the ripe pods, or yellow. So as we can see, for each of these characteristics, there is a pair. So he discovered that the plants, like I said before, either had this form of the trait or the other form. Okay, to better understand the observations and results of Mendel, there are some terms that we need to recall or to know, which I mentioned in a previous video. That is parental generation. The organisms or the individuals that make up the parental generation are the individuals that you bring and cross in that in your hybridization cross. Yes, those are the organisms you bring together, and usually they have contrasting forms of the same traits. For instance, you cross pea plants, so to say, that one of the parents is tall. Let's say this is the gene for height, and the other is short, tall and short. Okay, these are the parents. We call this the P generation. These are the individuals in the P generation. So if you were to, these are the gametes they will produce. Yes. So if you were to cross these two individuals, what you get is this. And these individuals, the offspring of these 2 individuals, are called hybrids or heterozygotes. heterozygotes. Okay. So P generation heterozygotes, or F1. Yes, you don't call them F1 individuals, you call them the heterozygotes or hybrids. Then there are different types of hybrids depending on the number of pairs of contrasting characters in the parents. We have monohybrids, dihybrids, polyhybrids. Okay, so monohybrids are hybrids that are gotten from crossing parents with just one pair of contrasting characters, like just maybe the gene for the character or the trait for height, like in the instance I gave. So just one pair of contrasting characters. For instance, let's say, yes, like I said before, height, tall, short, the same height trait but different forms. Now dihybrids are hybrids obtained from a cross that you are comparing the parents on two pairs of contrasting characters. Like in this case we have height, let's say red flowers. So two pairs of contrasting characters in the parents, that offspring are dihybrids. And polyhybrids from three pairs and above. Let's say here, what do I use as instance? Seed color. Okay, tall red flowers. Let's say constricted seed coats or inflated seed coats. Sorry, pods. So you have tall parent, red flowers, inflated pod structure or pod shape. Here you have the short parent, white flowers, and constricted pods. So these are three pairs of contrasting characters. Those will give you polyhybrids. Now the process of producing hybrids is called hybridization. And there are different types of hybridization crosses, which are according to what I named before. Monohybrid crosses, dihybrid crosses, and polyhybrid crosses. Polyhybrid crosses, depending on the pairs of characters, pair of contrasting characters in the parents, or depending on the type of hybrids produced. Okay. Now, back to Mendel and his work. When during his work, or in the course of his work, he noticed some characteristics or some patterns in the F1 hybrids. He noticed that when he crossed purebred parents, like I use the gene for height, this is purebred dominant or homozygous dominant parents, this one homozygous recessive parents for the traits of height. He discovered that when he crossed them, all the offspring were heterozygous, that they were hybrids, but they showed the phenotype of one parent, which is the dominant parent. That is to say that all the offspring were tall, even though inside them they had the trait for shortness. All of them were tall. So that made him propose the principle or law of dominance. Which states that in a pair of, let me not write it, which states that in a pair of, in a cross of parents with purebred parents with contrasting characters, only one form of the traits is expressed or appears in the next generation. That is, the dominant allele is the only allele that will be noticed or that will be physically expressed in the hybrids or in the offspring. Okay, that's it for the principle of dominance. And then he compared those characteristics, those 7 traits he noticed in the garden pea, to find out which one of them was dominant and which was recessive. And for flower color, he noticed that dominant and recessive. Okay, for flower color, you notice that purple was dominant over white. For flower position, you notice that axial was dominant over terminal. For stem length, you notice that tall was dominant to short. For seed shape, you notice that rough was dominant over wrinkled. For seed shape, pod color, he observed that green was dominant over yellow pods. For pod shape, you notice that inflated pods were dominant over the constricted pods. And then for cotyledon color, yellow was dominant over green. So this is what he established from his comparison in the garden pea. Okay. Now we go over to the F2 generation. Like I said in a previous video, if you were to cross individuals from the F1, you obtain the F2 generation. So when he crossed or self-fertilized individuals from the F1, he noticed that the trait that was recessive, that was maxed in the F1 generation, reappeared in the F2 generation in this proportion, 3:1. Let me illustrate how he saw that. This is the parental generation. Which gave us F1. And then when he solved this, we got these are the gametes for the male and and for the female. These are also the gametes. Okay, so you notice this. Get these two. Get these two. This and this. So he noticed that this is the F2 generation. So we have, sorry, yes, I've illustrated this. Let me just bring it in. So in the F2, we had TT and this. So he had 3 individuals out of every 4 plants displaying the dominant character, and then 1 individual displaying the recessive character, which was in the case of the F1 hidden. Remember, the recessive from this parent was hidden in this place, in this generation. But when we got to the F2, it resurfaced, but in this proportion. So this, it then proposed this as the monohybrid ratio, or the monohybrid phenotypic ratio. That is this hybrid phenotypic ratio. It is 3:1 from his monohybrid crosses. That's the cross that involves parents with just 1 pair of contrasting characters. So he discovered that the F2 showed this result in this proportion. That's some individuals, 3 individuals out of every 4 individuals showing the dominant character, and just 1 showing the recessive character that was, let's say, in the grandparents, if it were human beings. Okay, so at the end of his experiments, he was able to postulate he was able to give 3 postulates, which are as follows. 1, unit factors in pairs. 2, dominance, recessivity. And 3, the law of segregation. Okay, so he gave these 3 postulates, and I'm going to explain each one of them. Unit factors in pairs. He observed, or he said, that genetic characters are controlled by unit factors which exist in pairs in individual organisms. That's where we have these unit factors in pairs. Those are what we know now as genes, and these pairs, what we know now as the members of the pair, are called alleles. So they can be either identical or different, but they must always come in pairs. And most times, since he was working on a diploid organism, this applies to diploid organisms. That is, organisms with 2 sets of chromosomes, like humans. We have 23 pairs, as 46 chromosomes. So those are actually 2 sets of chromosomes. Okay, now dominance and recessivity. He said that unlike unit factors, that control the same trait or the same character, in a single, when they are present in a single individual, one tends to dominate over the other, and that one is called, the dominant unit factor is called, like I said, the dominant, what we now call alleles. And then the other, which is masked, is called the recessive allele. So that's for dominance and recessivity. When they are unlike unit factors, one is most times dominant over the other, and the one that is not dominant is called the recessive allele. Then for the law of segregation, this states that during gametogenesis, that is the production of gametes in sexually reproducing organisms. During gametogenesis, paired unit factors, those are those alleles, let's say this, paired unit factors separate or segregate randomly, whereby each gamete gets one of the two unit factors. That is, each gamete either gets this or this with equal likelihood. So it is either this or this. Both do not end up in the same gamete. So I think that would be all for this class. Come to the end of today's class. I hope that you've learned a lot today. In the next class, we are going to be discussing modified monohybrid ratio. That is what I just showed you. This 3:1 ratio is not always the case, but we are going to find out what causes this difference or these modifications. So if you like this video, please don't forget to hit the like button, share with your friends, and leave your comments in the comment section. Whatever you want, just let me know in the comment section. And also subscribe to our channel so that you can get updates whenever we upload a new video. Thank you very much for watching. See you next time.